really the sky's the limit. Like we have this database of Gal4 lines and UAS lines where you can mix and match. Yeah. You want males or females?
Well, maybe both.
Both?
Yeah,
Yeah.
we have it.
Yeah, we have an
What if we don't have a full hour?
Well, that's fine. Even if you just get a short video to use in your post-lab analysis, that's okay. It's really just familiarizing yourself with these methods. So when it gets to phase 2, you know, you can come in, you have the chambers already ready, and you know what you're expecting.
Oh, so there's nothing that's
The whole phase 1 is testing. Yeah. So, males,
Sorry.
females,
Sorry, sir.
and they're socially naive.
No.
So you just have to have a break in the
Okay.
Or these guys are good.
Dominant fly mine.
What was that?
One fly is
One
fly is dominant.
dominant?
Fight.
Is on the food cup?
No, the other one just stay away from.
Stay away from it? Yeah.
No, they haven't just food.
Where's the second one?
The
Somewhere hiding.
They're on the side.
He's right there.
It's hiding. It literally has been sitting there doing nothing.
Yeah, and you see the problem with social behavior.
Coward over here. What the hell?
So we have these Drosophila activity monitors that should be here anytime, well in time for phase 2, where there will be these small, it's not necessarily look like this, but there'll be hollow glass tubes, and you'll aspirate one fly at a time, and they go in the incubator in this system that automatically tracks the activity.
Very cool.
It's called the Drosophila activity monitors. The dams.
The dams.
Right. So it's either this week or next they come in.
Very nice.
Aspirating again?
What was that?
Aspirating.
Aspirating again, the
And
stress of
that's, you
aspirating.
see like all these skills you're building. Right? Lab 1, you work with flies. Can you sex them? Can you identify each of their developmental stages?
Has anyone ever swallowed?
And then,
Yeah.
oh, for sure.
Of course.
They're fine.
He didn't put the cod
You swallowed it.
Just extra protein. Yeah. Some diet, nutritional stuff right
As you
there.
guys are working on your independent projects, you know, we prepared those flies for you, but you don't have to stick with those ideas. Right? So basically, any Gal4 line we have, or any that you might find from another lab and paper, we can get, and we just need to cross it with one of the UASs, or many of them. Right? So let's say you found a paper that has a Gal4, you know, again, expressing in a subset of neurons in one part of the fly's brain that you think is involved in sleep, you think is involved in caffeine abuse, I don't know. And we get the Gal4, and then you can manipulate those neurons just by making 3 crosses, right? So you can developmentally silence them with TNT, you can chronically activate them with TRIP, you can get more temporal control by using Shabiri, by changing ambient temperature. When you test the flies at above 32 degrees, those neurons are off. You move those same flies to below 32 degrees, test them again, the neurons are back on. Or you use light. Red light, channelrhodopsin. Yellow light, turn them off, halorhodopsin. Okay, so you can mix and match the UAS effectors. So if I were you, I would choose, if you're gonna do something different, just choose one Gal4 and then make more crosses. Right, so you can control the activity of the cells.
Very cool.
Yeah.
Yeah, exciting.
Yeah, it's a lot of fun. Once you see the
for my
potential.
Yeah.
Yeah.
There's a lot.
It depends.
You just wouldn't be able to do that with mice.
It can be differential
Yeah.
based on
Once you
what
get
kind of
past the
activity it is.
idea
So,
that it's a small
let's say your
animal, but there's a
Gal4,
lot of potential.
from
Right, so
what
like, you know, you could literally have 6 crosses right here. One line, you're turning neurons on. One line, you're turning the same neurons off. Another line, they've been off since they were born. The other line, they've been on since they were born. And so you can really manipulate the activity of just that one cell in different conditions. And then see what the effect of the behavior is. It's pretty
They'll
cool.
walk backwards, they'll jump around the plate when you shine light on them.
So how
So
long
some
are your
behaviors
recordings?
are
20
visible.
minutes?
Other
Okay,
than not
so this is
some,
good,
like,
unless we want to
the
keep
flies that I
recording.
do research on,
Because we have
they
7
don't
minutes on that
exhibit
one and
a
13 on
behavior
this
that's
one.
visible.
So what do you guys think?
is not visible. We don't use light mediated.
Does it leave them? Yeah, might as well. I mean.
Want to put two different ones? Want to put two different ones in there? And try it again?
Worst case,
Yeah.
we have these. Record these
Okay.
two animals. So you have something to see. Right? So worst case,
These two are not
depends on
moving.
recording those.
Neither. None of them are really doing much of anything, quite honestly.
So like
There we go.
the all cholinergic neurons, right?
Should I time
Theoretically,
this too?
the behavior that
You can.
leads to see.
We had one group do that last year, so.
Did they activate it with light?
With red light, they call it, the over seas.
Yeah, exactly. So some of them you turn it off.
The problem with, so the channel rhodopsin UAS, this CS Vincent, is called it's leaky, which is basically, so this one,
They're just
it's
further
leaky, so
apart.
it's not time locked. So unfortunately.
I just wanted to
when you
cap
activate
two
the
more
cells,
flies.
They stay in a
You
depolarized
start out at
state.
the beginning, you're like, oh, I feel so bad.
And now I'm like,
you're
I wanna do
wanna
it.
Do it.
No, but
Wait, let's
eventually,
decapitate this little
depending on
bad boy.
The cells that they're expressed in, rhodopsin,
Yes,
um,
we're good.
It'll come back to resting state, the resting membrane temperature. But there are other crimson, channel rhodopsin lines too that are more time locked, but they use those for electrophysiology. So can you imagine, there are labs that do electrophysiology in the fly brain. Electrophysics, single cell. Wow.
Look like a nano or like a
Pipette. Wow. Yeah. No. It's a, but see, you can see how that design is incredible. So you have this whole toolbox. These are just a few. There are thousands.
Oh,
of
okay, we got some
lines
good content
where
here. Some good content. They're coming really close to each
that
other.
are publicly
Really?
available for very low cost.
Took
Each
them
of those
long
lines
enough.
costs about $10. And you can, because of the balancer chromosomes, you can keep flies for many generations. You don't lose the chemistry. So, yeah, there was a really famous woman, Rachel Wilson, at the next door when I was at Harvard Medical School. She won a MacArthur Award because she pioneered electrophysiology through flies. Yeah. So
Oh,
the beauty
good
is you can
content.
use these really exquisite tools. So now you're doing electrophysiology, you're patching onto one neuron, and then you're controlling it with ambient temperature. You're controlling it with pulses of light instead of electrical. That's all through, you have this set of goggles that can multiply by 5 times. Yeah. Exquisite. It's elegant, these techniques.
Does it count if they're doing stuff if they're not around food?
I showed.
They're interacting.
And they have negative.
Yeah, look at them. They're right next to each other.
I still want to fight. I don't fully understand why. Because theoretically, the gene that it was married to was the scabbard gene. So I didn't
So
understand.
weird,
But
they're
there did say there was something in it.
You signed up for it.
I remember you mentioned that for some reason.
The
they
game
negative
footage.
Hmm?
The game footage.
Okay.
Tell me wrong. You come in, right?
Because I was, I remember from back, way back. I was curious if that has to do with me. If it has to do with me.
The flies that have more balancers.
Approach two.
It's like a headless chicken. Oh my God.
So the more that transition.
I feel like that's true for most animals. Yeah. They're not meant to exist like this.
So behaviors that are natural, like
Yeah, so we could always try and get somebody to somebody else's. I mean, we don't really need it.
so flies are also going to attack.
Well, I just realized that right now. It's okay. Yeah. I mean, I'll just
this just in case we need another chamber.
About, let's say there's a light emitting from the bottom of the tube, not at the top of the tube.
I really like that.
Alright, it's doing something, but what is this?
Theoretically, it was darkened around the top. But the fly is still coming towards the top of the cave, and if you well, there was light in the bottom.
Well deserved.
Is that their behavior? What are they doing?
Oh, let me see. What are they doing?
Okay, I was wondering if there was.
There's a hierarchy.
That's gotta count as something.
Oh yeah, for sure. That's definitely